will be able to catch up.
Okay, well, welcome everybody. I'm very happy to introduce Dr. Joe Harding. He's Emeritus Professor from the College of Veterinary Medicine. I'll let him give you the other details, but he has been a long-time researcher at WSU, and I knew him and worked with him off and on over the course of the years that I was in the OGRD. So, Joe, would you like to go ahead and get started? And
Sure.
Bill, any other information you'd like to put in?
So, first of all, I'm gonna share a screen here in a second. As Nancy said, I was at WSU for 45 years, and have been at the College of Veterinary Medicine that whole time. During that time, I've been collaborating. My research has been in collaboration with Jay Wright, who retired from psychology about 5 years ago. He was the chair and many other things. My best friend, my collaborator. So everything I'm going to talk about today is really stuff that Jay and I did together. And I have a picture of Jay in one of the slides here. So what I want to talk about today is the core work that we started right around 1990 that ultimately led to the formation of a biotech company in Seattle, or in Bothell, that now has multiple drugs in clinical trials, some of which are designed for treatment of dementia, specifically Alzheimer's disease. And I don't have slides related to that on here, but I expected that people will have questions about that at the end. And I will tell you what I can tell you about the clinical trials and where things stand. So I'm going to share a screen here and get started. Let's see. Hopefully this will work. Can people see that? I'm assuming you guys can see this. Did I see yes? Yes. Okay. So let me see if I can get this. Let me get in the right place here. Okay, now we're good. So I want to talk about a group of proteins that are called growth factors. And the way you can think about growth factors is these are your executive proteins. They control everything. And there's a relatively small number of those. And it's been very difficult over time to develop effective drugs that modify their activity either up or down. You could imagine, because these things are so important, if you could manipulate their activity, you really could actually impact almost any disease entity you could imagine. Okay, so just to give you an idea of the kind of things that growth factors are involvement, all of embryological development is totally orchestrated by growth factors. Our maturation to adulthood is totally mediated by growth factors. The immune system, many of these drugs that you hear, you know, the Enbrels and things like that you hear advertised on TV, all of those are affecting growth factors that impact the immune system. All of these things for psoriatic arthritis or eczema or other things, that's what they are. These are, but they're all what are called antibodies, and we'll talk about those a little bit later. Growth factors are also totally important for central nervous system development. Day-to-day activity, including learning and memory, are totally dependent upon the availability of growth factors. And what we're really interested in is growth factors are necessary for maintaining the health of the nervous system, specifically the brain, and they're involved with repair. So when the brain gets damaged, growth factors get turned on, and a couple specifically get turned on. And one of those is the growth factor that we work on. So again, why would you develop drugs to alter growth factor function? It turns out that moving them up or down is beneficial to many different diseases. For example, in human cancers, which I'm not going to talk about today, although I have plenty of data that I could show you or insights I could talk about regarding growth factors in human cancers. Every human cancer that you've ever heard of has at least one growth factor system that's overactivated. Every single one. Some of the worst growth factors are ones that are called survival factors. These make it very difficult to kill the cancer, for example, with chemotherapy. It turns out that the growth factor we work on happens to be a survival factor. So inhibiting its activity has anti-cancer activity. Like I said, that's not going to be the focus of our talk today, but I could give you a whole talk just on the inhibition of these growth factors. The flip side is there are lots of instances where you could benefit by augmenting growth factor function. Whenever you have any kind of injury in the brain or in many places, this turns on growth factor function to repair it. And if you can rev that up, you can improve that repair mechanisms. And that's the basis for the drugs that we develop. As that you can see here, there are almost any disorder you can think of could be impacted by improved growth factor function. As I mentioned, you could inhibit growth factor function, and this might have some effects that are positive with regard to cancers, or you could activate it in cases where you need regenerative capacity. Currently, there are no drugs, none whatsoever, that are available to activate a growth factor. The first ones are the ones that are in clinical trials at our company right now. There are drugs that can inhibit growth factor function. These are, as I said, are almost all antibodies that are designed to interact with that particular growth factor or its receptor, that is the cellular site that the growth factor talks to. The problem with these drugs is they're insanely expensive to make. A typical cost of an antibody for treatment of some disease, specifically, let's say some form of cancer, can run a patient well over $100,000. They're just not sustainable as a means of inhibiting growth factor function for the greater population. It may be fine for wealthy countries with wealthy people, but it's certainly not going to do anything for people in Bangladesh. So finding cheaper ways to block these growth factors is a laudable activity. So how did we get interested in growth factors? Luck. That's how we got interested. Jay and I, this is where we used to meet at the Cub every day. And this is, I can't remember who took this picture, but every day Jay and I would have tea, and Jay would write my idiot ideas on napkins. He had a whole drawer of them, hundreds, if not thousands of napkins with my weird ideas, most of them that were useless. But every once in a while, there'd be one that was worthwhile. And Jay would help me decipher what was useless and what was not useless. But anyway, we worked on a series of peptides called angiotensins. And the name, if you listen to the name, angio means blood, tensin is pressure. Angiotensin 2 is a peptide, which all a peptide is a teeny protein. And this peptide has been implicated in regulation of blood pressure. It increases blood pressure. One of its breakdown products is something called angiotensin 4, right here, okay? And we discovered that angiotensin 4 had a site that it interacted with in a number of tissues. And what was startling at the time is angiotensin 4 was considered to be non-active because all it was a breakdown product of angiotensin 2. So nobody paid any attention to it. Well, we discovered that there were these binding sites that angiotensin 4 stuck to. And one of the methods we used is shown here. This is called autoradiography. Basically what we did is we took this molecule angiotensin 4, and we put a radioactive tag on it. And then we took different tissues and we sliced these tissues very thin, put them on slides, and we incubated them with this radioactive angiotensin 4. And we asked, hey, where does it stick? And the reason we were doing this is if we could see where it stuck and we knew something about that site in terms of function, we would have an idea of the function it might subserve. So this is a section through the brain. Obviously, this is a section through the heart. Everything you see here, all the darkness, that's where angiotensin 4 is binding. This little area right here, this very dark line here, this is an area called the hippocampus. This is the area of the brain that integrates new information, amplifies it, and puts it out for long-term memory. These are the areas that die in Alzheimer's disease. These are actually nerve cells that are staining here. These guys die during the development and the progression of Alzheimer's disease. So when we saw this, we said, oh, geez, I wonder if this has something to do with cognition, learning, memory. And lo and behold, it did. So over here in the heart, this is cardiac muscle. It has huge effects on cardiac function. Something we would like to work on in the future. These are blood vessels over here, and the lining of the blood vessels are called endothelial cells. And what they do is they're important for regulating the ability of blood vessels to sprout and vascularize new tissues, a process called angiogenesis. So we, just by looking at this, we thought, hey, this might have an effect on cardiac function, and maybe it has effect on blood vessel vascularization. It turns out both of those things are true. So this is how we homed in on, hey, what does this new small peptide called angiotensin 4 do? So I'm going to skip ahead a little bit, and we actually did a bunch of work, and we tried to see, is there anything that looks like angiotensin 4 in the literature? And it turns out that angiotensin 4 looks like a piece of something called hepatocyte growth factor, a little teeny piece. And it turns out that the piece it looks like is the most important piece for the regulation of this growth factor's function. So this was just serendipity. This was luck. But we realized this thing is like, sorry, let me go back here. I don't know why it's going forward by itself. So this, we noticed the similarity between angiotensin 4 and a piece of HGF. And then we started asking the question, well, okay, if they look a lot together, if they look similar, do they have the same functions? Do they do the same things? And here's a list of things. Both of them were shown to be involved with learning and memory. Both of them provided what's called cerebroprotection. This is protection against toxins. Both could stimulate nerves to branch, nerve cells to branch. They both could induce blood vessels to branch. They could turn on stem cell function. They both had pro-wound healing effects. So when we saw this list coming together, we thought, you know, we really are on the right track here. This probably is a molecule that is interacting or acting on HGF. This is HGF here. You don't need to, this is the general structure of it. But HGF is one of these things I mentioned earlier, they're called survival factors. Very potent. In fact, it's the main repair growth factor in the human brain. So it's the perfect target to go after if you want to improve recovery from brain damage, be it from a traumatic injury or from a disease like Alzheimer's, Parkinson's, ALS, whatever. This growth factor, which is a huge protein, interacts with cells through a receptor protein. So you can imagine like a lock and key. You've got HGF, and when it has to interact with a cell, it interacts by binding to a specific receptor protein. This thing happens to be called MET. And what we know is that the activation of HGF with MET does a whole bunch of things. It stimulates cell proliferation, cell movement, changes in cell shape, but most important for us, it increases cell survival and repair. So I mentioned that the angiotensin 4 molecule looks like the most important regulatory site on HGF. And this regulatory site sits right here. It's called the hinge region. And it's a 6 amino acid. Don't worry about what these are abbreviations for different amino acids. There's 6 of them here. And it has some similarity to angiotensin 4. So basically, so at this point in time, we said, hey, we've got this angiotensin 4. It looks like it activates this growth factor, which is critical for many things. Could we actually go in and build drugs around this sequence? I'm a medicinal chemist. That's what I do. I build drugs. So we wound up building several hundred analogs, things that looked like angiotensin 4 or looked like this hinge region. And we started screening them. Some turned out to be antagonists. They blocked the activity of HGF. These became anti-cancer agents. Some were activators. These became repair mechanism type molecules. What I'm going to focus on today is I want to talk about the activators. I actually have a little antagonist stuff at the end, but I didn't want to take up too much time. So the activators, these are the guys that are the molecules that are effective at repairing the brain following damage by neurodegenerative disease. So here's what we know about HGF and neurodegenerative disease just from the literature. We know that in all of these diseases, Alzheimer's, Parkinson's, ALS, MS, spinal cord injury, the levels of HGF go up. And you might say, geez, that's a bad thing. They're going up with the injury. What it really is the body's response to injury. You've got an injury, so it's revving up the repair mechanisms. One thing that's really interesting is if you look at MS, there's 2 forms of MS. There's a form in which you have bouts of MS, and then you have remission. And then there's a type of MS that's called progressive, where there's no more remission. This is terminal MS. And if you look at the levels of HGF, what you find in the early stages with activating and remitting MS, you see elevations of HGF. But once the person switches over to this unrelenting progressive MS, for whatever reason, HGF is no longer can be elevated, which means that repair mechanism, which was supporting the remission, is lost. If you look at animal models of these diseases, of different forms of dementia, Parkinson's, MS, ALS, spinal cord injury, in every animal model, if you artificially put HGF in there, it has positive effects on the outcome of the disease in these models. So naturally, it goes up during these diseases. Artificially, if you add HGF to disease models, it improves function. So this told us that targeting HGF for neurodegenerative disease was a great target. So as I mentioned a second ago, we built hundreds upon hundreds of molecules, but we wanted to build something that would be a drug. So what makes a drug, especially a drug that's going to work in the brain? So there's several things that we had to accomplish with the chemistry. And I'm not going to show you any chemistry here, so don't start freaking out if I'm going to put a bunch of chemistry up here. I'm not. So the first thing is we wanted it to be potent, which means it had to have a high affinity. It had to be really sticky to HGF. And what this means is we can use very little of it. And one of the reasons when you're developing a drug and you want to use very little is most drugs do other things that you don't want them to do. The lower the concentration, the lower those side effects. So we wanted something that was super high affinity for HGF, and we achieved this, astronomically high affinity. We wanted something that was as specific as possible. We were able to achieve that. From a practical standpoint, the difference between, say, angiotensin 4 and one of our drugs is if I injected you with angiotensin 4, it literally would be gone in 15 seconds. It's very metabolically unstable. So it's not going to be a drug. So we had to incorporate into the structure changes in structure that would bypass the normal mechanisms that would degrade the molecule. The next thing, and this is the toughest thing of all, your brain is protected by something called the blood-brain barrier. It makes perfect sense when I tell you about it. The brain is your most important organ. You don't want things willy-nilly going into the brain. So if we actually took a close look at a blood vessel in a brain blood vessel, what you would find is those endothelial cells that I mentioned briefly that are involved with angiogenesis. In tissue outside the brain, many of them, there's a lot of spaces between the endothelial cells, so things can slip through. The brain vasculature, they're jammed right against one another, what are called tight junctions. Nothing gets between the cells. In addition, there's a layer of additional cells on top of it. So it's very difficult to get things into the brain. You can trick that system by making your molecules look a little bit more fat-like, so they can dissolve right through the membranes of the cells. So we did that. Now, you have to keep in mind, doing that, we had to maintain its metabolic stability. We had to maintain its specificity. We had to maintain its potency. So these things are all interconnected. You can't just change one and destroy the other properties. Every property has to be incorporated. The two other things we wanted to have a drug that could be taken by mouth. It turns out we have forms that can be injected and or taken by mouth. But ultimately, we wanted to have an orally active form so it would be easy to take. And the last thing, and this was thinking about patients, we didn't want something that was so expensive to manufacture that no one but the rich would be able to use it. So we had to design something that in the end could be manufactured relatively inexpensively. Our lead drug that's in the final phases of treatment of trials for Alzheimer's, a single dose costs $1.34 to make. So that was the goal, and we achieved that goal. So I want to show you a little bit of data. How do we know these things work? What told us that we were on the right path? I showed you a whole bunch of lists of things that they did, but I wanted to show you a couple videos, and you guys may have seen these. And the first video I want to show you is a video that's designed to test an animal's memory, okay? And it's something called the Morris water maze, okay? And the water maze is interesting. It's a tank of water filled with water, and sitting just below the surface of the water, you have a pedestal, a hidden pedestal. If you put a rat in here, they can't see the pedestal because it's below the water level. But around the tank, we have visual cues so that they can orient. Once they know where it is, they can say, oh, it's close to the green circles or the blue turquoise circles here. So they learn very quickly how to orient. For example, if you look over here on the right, this is an example of a test where you put the animal in. It was put in right down here at the bottom. It wanders around. Eventually, it finds the pedestal. The more often the animal's put in here, the faster it gets. After 8 days, 5 trials a day, this animal will beeline right to here, okay? So they get, each test is 120 seconds. Between tests, they get 30 seconds and they get 5 trials a day. So they're in here a total of potentially of 600 seconds. Like I said, at the end, an animal is in here for 15 each trial. So they're not even in here for much more than a minute at the end of 5 trials. When Jay and I first set this up, and this was after we discovered that autoradiography picture that I showed you, where we had the binding to those areas of the brain that are involved with learning and memory. Neither Jay nor I were doing learning and memory, but we said, well, we need to do it because that's what the autoradiography is saying. So we set this up. When we first set this up, I went over to the grain supply and I bought a horse trough. Okay, that was going to be we built a pedestal, and this looks very scientific here, triangles, squares, circles. The reality is, when we set this up, this was Magic Johnson, okay? This was Larry Bird, okay? And this was Michael Jordan over here, posters. So we had posters of basketball players around. On the floor, we had an X where the experimenter stood, because they also orient to you. As we got more sophisticated, we got rid of the posters and actually used something that was a little more scientific looking. So I'm going to show you, it takes a second to come up. I'm going to show you a video. So what model do we use? We've used lots of models. The best model is the aged rat. Just like people, if you're at 85, you have a 50% chance of having some level of dementia. A 22-month-old Sprague Dawley rat has a 50% chance of having dementia. So rats are very much like us. So what we did in this study is we took a bunch of aged rats, and you can't even imagine how expensive it is to buy these rats. They're like $350 a piece. So we bought almost 100 of them, okay? And we screened them in one test to find which of the rats were demented and which of the rats were normal. We discarded the normal ones. So we threw out $350 a piece of half of those rats. We took the ones that were left and we said, okay, let's test them in a novel learning environment, the water maze that I just showed you. Half of them were treated with drug for 3 weeks and half of them were not. But you remember, all of them showed dementia before we started this. So you're going to see two water mazes here. Oh, it's weird. Let me go back. Oh, sorry, down here. It takes a second to come up. Okay, two water mazes. So this is the tank I showed you. This is a new tank. These the little orange and black things, these are heaters. So I'll play this again for you. Remember, both of these animals were totally demented before they started treatment. The one on the right had treatment for 3 weeks. The one on the left, placebo. So I'm going to go back here and start over again. So I'm going to pause this, go back here. So the rats are placed in the water maze. And then remember, the maximum time is 2 minutes. Here's what happens to the treated animal. So here's the treated animal going in. It's learned this thing and it's going to beeline right there, makes it there in 5 seconds. On the left, same demented animal, it just didn't get 3 weeks of treatment. To make a long story short, this guy's going to be in here for 2 minutes, okay? And what happens at the end of those 2 minutes, the typical procedure is the experimenter, in this case, one of my graduate students, Jewel, is going to pick this animal up and she's going to place it on the pedestal so it has another opportunity to look at those external cues. So I think she's going to pick it up here in a second. Where is it? There, she picked it up and, oh, here she's going to pick it up. Here she comes. So she's going to pick it up at 2 minutes or thereabouts, stick it on here, it gets 30 seconds. The point here is that these animals were the same before they were treated. The one on the right was treated for 3 weeks with drug, the one on the left, 3 weeks with placebo. The actual data looks like this. So we're looking at time, training day time. So this is what a normal young rat does. Remember, 5 trials a day. This is the mean of the 5 trials on day 1, day 2, day 3. You can see by day 8, they're on this, right? They're right on it. This is what an aged, untreated animal. it never, ever learns it. Yeah, it may improve a little. Actually, these are not statistically significant from one another. So statistics say there's no change. This is the same group as this, but treated. And they're not quite as, they're not a brand new young animal, but by the time they get down to day 8, these guys are actually not statistically different. They're a little slower getting down there. They don't come down as fast, but at the end of 8 days, I'll talk about this at the end when I talk about clinical trials. 8 days turns out to be an important date. Okay, so that's one disease we went after. Another disease, and we have a trial running right now called a SHAPE trial, which is a Parkinson's trial. It started in January. And one of the other impacts we saw with these drugs is that they can affect symptomology in Parkinson's models. What I'm going to show you here are 3 rats, and they're going to be performing what's called a rope hang. They basically hang on here, and we time how long they can hang on the rope. If we induce a Parkinson's model, what we find is that if we let the model develop after about 10 days, so the model's induced, it gets worse over about 10 days. And by 10 days, a typical Parkinson's rat can hang on this rope up here for about 5 to 6 seconds, okay? So I'm going to show you 3 rats here. I'm going to show you a Parkinson's rat. This guy has been tested all along. At 10 days, it hung on for 6 seconds. Now we're out 34 days. And during that period of time, it's gotten a placebo drug. This guy over here, at 10 days, it was 6 seconds. It got treated with drug to day 34. This last one called naive, this is just an age-matched normal rat. What does a normal rat that's the age of these 2 that you're going to see here do? And so I'll start this. You'll see one of the students stick the rat on. So this is our Parkinson's rat here. It was 6 seconds back at day 10. It's still 6 seconds at day 34. Here's a normal rat over here. Remember, this guy was 6 seconds at day 10. He's been treated for 34 days. One of the side effects of this drug is it increases muscle strength. So the difference, this is treated 5, 6 seconds, normal 15, treated 34. One of the FDA saw this data. This drug was designed to be given orally. They were very worried about it becoming a drug of abuse for athletes. So we formulated for injection. So we're injecting this drug in patients specifically to make it harder to deliver, because the FDA was very concerned about this data and that athletes would be using this to increase their muscle strength. This is what the data looks like. Again, this is our normal rats here. Just they get better over time. They learn. So this is just normal controls. Here's the guys. I guess I said 10 days. This group here was actually at, I guess, day 15. We started treating them. But what you see is this is what happens with placebo. This is what happens with treatment. As I said, we have a trial running now, a Parkinson's trial running. It's based primarily in Seattle. So how does this work? How do these drugs, how does HGF do this? What could it do to improve function in these animals? The first thing it does, and this is probably the biggest of all, is it turns on something called synaptogenesis. It's a big word, but all it means is that it allows nerve cells to make new connections. One of the common properties of every neurodegenerative disease that you know in the beginning, the first thing that happens is the nerve cells in whatever area of the brain is going to be impacted start to lose connectivity. They become isolated from one another. So that makes it much more difficult to get information flow. It also, another thing that happens when nerves interact with other nerve cells is not only does information flow, but growth factors are released from one neuron, they go to the second neuron and they keep it healthy. So once you lose those connections, not only does information flow go down, but the ability to maintain the health of the secondary neuron goes down because that connection between the primary neuron and the secondary neuron has been lost. So our drugs are incredible at stimulating nerve growth. And I'll show you some data in a second. The second thing it does is it stimulates the production of new neurons in that area of the brain called the hippocampus that I mentioned earlier, where I mentioned that it's the integrative center of the brain. One of the things that you should probably know about the hippocampus is you can imagine a lot's going on there all the time. Every time you see every time you experience something new, hear something new, see something new, that information goes into the hippocampus. It's churned around, amplified, and sent out to memory. The hippocampus uses more energy than any other area of your brain. And as a result, just the generation of energy generates toxins that kill those neurons over time. Now, most of you have probably heard that, nerve cells, you only get them once, you never replace them. Well, the hippocampus is an exception. We have what are called stem cells that sit there that are replacing neurons all the time. In fact, if we didn't have them, we probably would have all been dumb by the time we're 4. So we're replacing these guys all the time. Now, in neurodegenerative disease, that replacement activity goes down. So not only are you losing connections, but in this core area of the brain, we're not replacing neurons as fast as we should. HGF and these molecules stimulate that process. The third thing they do is neuroprotection. As I just mentioned, the more active a cell, the more toxins it produces. Some of you have probably heard of oxidative stress or oxidative damage. This is just normal damage that happens because you produce molecules that are very reactive in your mitochondria during energy generation. There's a whole set of protective mechanisms to get rid of these. HGF and our drugs ramp up those mechanisms. And you might say, well, why does it do all 3 of these things? All 3 of these things are integral to repair. This is a repair growth factor. So it's going to have its fingers on everything that would be important for repair. So Nancy, you probably should let me know how I'm doing my time. I'm not watching very carefully here. Are we okay still?
We are okay still.
Okay, so this is just to give you an idea. This is a picture of the Parkinson's model. And what we're staining here, this is the area in the back of the brain, the midbrain, in which cells die during Parkinson's disease. It's called the substantia nigra because it's nigra because it has black pigment in it. So what we do in this model is we actually artificially go in and we lesion with a chemical that mimics the oxidative stress I just mentioned, that mimics that, but in a big way. We injected it in one side and we actually destroyed those nerve cells. And then we stained them to see who's left. Okay, so this animal has Parkinson's-like symptoms. We did the same thing here, but the difference was this treated animal here had one of our drugs. And what you can see is this site is restored to function. I mentioned the growth of branches from nerve cells. They're called dendrites. And what we're looking at here is these orange strands here. These are dendrites, and all these little kind of yellow, white dots on them, Every one of those is what's called a synaptic connection. That is where one nerve cell is talking to another. You can imagine to have connectivity, there's two ways you could have connectivity. You could cause this tree here to branch more, or you could increase the number of individual contacts on each branch. These are two different drugs. This is one is related to angiotensin 4, and this is one of our newer drugs. In fact, this is a modification of this drug is in clinical trials right now. So this is what a control looks like. This is what an angiotensin 4 drug looks like. This is what a mature drug looks like. This is 7 days of treatment. It's unbelievable. In fact, we see significant increases in what's called arborization, the branching, and the production of these spines where the connections are made. We see visible and statistically significant changes after 30 minutes of treatment. So what are the potential use of these activators? It's a long list, right? Every one of these is probably a good target. We actually have data in animal models with ALS models that suggest that it's going to be useful there. We have data in MS models saying it's going to be useful there. As I mentioned, one of the side effects of this drug is increased muscle strength. Not only that, but it increases the production of new muscle fibers, something that's lost in boys with muscular dystrophy. So we haven't explored this, but it's clearly something that should be looked at. We've looked at stroke models and recovery from stroke models. No big surprise, this improves that. We haven't done any traumatic brain injury, but this is a no-brainer. This is something we should be looking at. In fact, Athera plans on looking at this. We also haven't looked at spinal cord trauma. That's something we plan on looking at. These molecules, and we have really spectacular data, are amazing at wound repair, even in diabetics. So this is something that is under development. There's some reasons to believe that type 2 diabetes, I'm not going to go into the details here, would be amenable to treatment with these drugs. And almost any disease called fibrotic disease. What are fibrotic diseases? These are diseases in which the main cells of that organ are being replaced by fibroblasts, and fibroblasts are non-functional. They just produce connective tissue, collagen type things, cartilage type things. And this typically basically pushes out the important cells that do the job. In the heart, it would be the cardiomyocytes, the muscle cells. These would be the lung epithelial cells, and different places, different kidneys and liver. We actually have some data suggesting that these drugs are going to be very effective in different fibrotic diseases. Okay, so I have some more stuff, some bonus stuff, but I thought I'd take a break and tell you a little bit about the clinical trials that are going on with regard to the neurodegenerative disease. So what we've completed, so what Athera has completed, they've completed one set of clinical trials. Let me tell you about clinical trials, first of all. How are they organized? Clinical trials typically are called phase 1, phase 2, and phase 3. Phase 1 is typically just, there's two questions you're asking in a phase 1. Is the drug safe? And typically it's done with volunteers who are not patients with the disease. The second thing you're asking is something about what's called the pharmacokinetics. How fast does the drug get into the circulation? How long does it last? Basically, you're trying to get an idea for how effective is this drug going to be and how long is its effect going to last. And this is done by taking serial blood samples, or in the brain, you might take serial excuse me, serial samples of cerebral spinal fluid. So that's typically what happens in phase 1. Sorry, I'm gonna take a cough drop here. So in phase 2, so phase 1 typically has anywhere from say 20 to 40 people. Phase 2 could have, it could be up to 200, it could be less than. In phase 2, you're going to start to look at safety issues with patients, and you actually might start to look at, hey, does it actually do anything, right? In phase 1, you don't even ask that question typically. If the data, if this preliminary, what's called efficacy data looks good in phase 2, you go to phase 3. This could be a trial anywhere from hundreds of patients to thousands. And you might say, well, what determines how many people you use? It's how robust is the effect? If you have a huge effect, you don't need very many patients to demonstrate that this works. If you've got a very modest, very marginal effect, you might need thousands to see this statistically. Yeah. So the first trial, so one of the first things we did that was different than almost any other biotech company is we asked the FDA, based on the data I showed you, this spectacular data that I showed you, could we combine phase 1 and 2? And could we combine phase 2 and 3? And what we did is we split phase 2 in half. And we called our first trial a 1B/2A trial, and the second a 2B phase 3 trial. Something that no one ever does. In our first trial, we used patients, Alzheimer's patients. The numbers were very small. I think the trial had like 16 patients in it, okay? And so what did we ask in that first trial? We decided with so few patients, we're not going to ask memory questions. The variability is too great. We were going to ask something objective. And what we did is we measured something called an ERP, an evoked response potential. Basically, what you do is you stimulate the brain in some way. We used a tone. The patients had earphones on. We gave them a complex tone. And then we had an array of electrodes on their head, on their scalp, and we could measure how fast that information that went to what's called the auditory cortex, which is linked to the ears, to the rest of the brain. We could measure that speed. We could measure its robustness. And no big surprise, in Alzheimer's patients, as the disease progresses, that speed gets slower and slower and slower. With advanced Alzheimer's disease, it can take 3 times as long. And when you see the signal over here, it's massively degraded. So it's no big surprise that Alzheimer's patients have cognitive problems. Okay. So we measured this. It's called a P300 measurement. And we did this P300 measurement, and we had our Alzheimer's treated patients, and there were about 8 or 9 of them, and we had our controls. There was about 6 or 7 of those. When we started out, sorry, when we started out, all of the patients, all of these Alzheimer's patients showed an ERP which was much slower than normal, way slower than normal. We treated these guys daily. By day 8, every treated Alzheimer's patient was normal, completely normal. The placebos, they continued to degrade over that 8-day period. That's why I said 8 days is a critical thing. So when the FDA saw this, they cut us a bunch of breaks for our phase 2B/3 trial. They said, well, this effect is so massive. There was not a single Alzheimer's patient that didn't improve to near normal levels. And there wasn't a single placebo that improved during that time. They said, you don't need thousands of people. So our trial that's running right now is somewhere between 300 and 400. It'll finish up mid 2023. And you might say, why does it take that long? It takes a long time to recruit your patients. There's certain criteria we had to have to get everybody to be similar to one another. We didn't want our patient population to be so diverse that we could never make a decision about the effectiveness of the drug. So we had criteria that they had to fit. It takes a long time to recruit 400 patients. We started in 40 different centers around the world. We've honed that down now as we started to basically go in and evaluate the different centers to see how well they're doing. And that is probably down to about 20 right now. About half of the patients now have finished the trial. One of the things that we are able to do is the trial lasts 6 months, they get drug for 6 months. What we're able to do is those patients then, when they finish the trial, they can continue taking the drug. And it's called an off-label extension. And there's been some issues about the off-label extension in terms of whether some of the people in the company didn't want to do it, but ethics and morals say you have to do it. And we are doing it. The demand from the patients, caregivers, neurologists was almost universal, meaning the drug is working on these patients and they absolutely want to keep taking it. The official data won't be unlocked until around July of next year. If the data mirrors what we're seeing with individual patients who are reporting to us as individuals, not as analyzed data, the drug will probably be on the market in 2024. That would be the expectation if things continue the way it's going right now. If you know anybody who has Parkinson's dementia, we're actually recruiting. We're still recruiting people for the Parkinson's trial. So if you know anyone, tell them to go down the WSURA site, look at clinical trials, and there'll be information for recruitment. We aren't recruiting any Alzheimer's patients right now. That's all done. So that's kind of where it stands. I can stop there. I threw a couple slides in if you guys were interested in the flip side. I've basically used up, it's 2:26. Like I said, the antagonist of HGF is a tremendous anti-cancer agent, absolutely phenomenal. And this is not actively being developed right now, only because we're a small company and we can only do so much. This is like sitting on the back burner, this data. So I don't know if you guys want to hear this or not. I can stop right there and take questions.
If anyone has any questions at this point, go ahead and unmute yourself. Or you can also type it into the chat.
Yeah, I guess I can use chat. There's no dumb question. I know I covered a lot of things. I tried to explain things. I hope at a level that people are not trained like I am understand.
Joe, this is Nancy.
Okay.
In all of this, did you have an aha moment, or is this just a natural progression of discovering about the HGF and that?
So there were two aha moments, okay? The first was, I personally, I didn't tell you the whole story about how angiotensin 4 was discovered. We were working on something else. And I came across this peptide and found out that it had binding sites in interesting places. That was an aha moment, okay? The second was Judah Folkman. You may know the name, he's a Nobel Prize winner. He was an oncological surgeon, and he made this discovery that people that have surgery to remove a primary tumor, often 4, 5, 6 months later, all of a sudden, after he's removed the primary tumor, secondary metastatic tumors start popping up all over the place.
Ooh.
And what happens is that these little what are called micrometastases, they're actually inhibited from getting their own blood supply. The primary tumor puts out a molecule that blocks vascularization, blocks this thing I called angiogenesis, okay? And it blocks angiogenesis so that basically the only blood supply is occurring to the primary tumor. All the daughters are starved. Soon as you remove the mother, that anti-angiogenic factor is gone. They all stimulate their own blood supply, and then you've got metastatic tumors everywhere. Okay.
Yeah.
What I'm getting to is he discovered what this molecule was, something called angiostatin. And angiostatin belongs to a family of 3 proteins, something called macrophage stimulating protein. It's a piece of plasminogen. And the third one, hepatocyte growth factor. And when he published this paper in Cell, and he had the structure there, what turned out to be the hinge region looked almost exactly like angiotensin 4. So I'm reading this paper, and I look down and I see our structure in the middle of this. And we knew that we had angiogenic effects already. So then I said, well, probably we know these other effects. This is a family of 3 proteins. Which one would be the likely target? Hepatocyte growth factor. So that's how things work, you know. When we first started working on it, almost nothing was known about it. So they were the 2 big aha moments. And there were huge milestones during synthesis of drugs that were made. You know, when we finally got it across the blood-brain barrier, I mean, I can't tell you the thousands and thousands of hours that went into doing that.
Wow.
But when we were successful for that, we knew we've got a drug.
Wow.
So that, not exactly an aha moment because it was thousands of hours to get there, but we did it. We've got it. So they were probably the 3 big things.
Wow. Christina, I think we have some chat questions, I see.
Oh yeah, let me go over here and see what.
Yeah, I see 3 of them, Joe.
Okay, let me open them up and see what we have here. Okay, so there's a question about, they know someone who has Parkinson's, where can they get more information? So the company is called Athira, A-T-H-I-R-A. If you go to their website, they have a pop-up menu for clinical trials. Go to that clinical trials and look for what's called the SHAPE trial, S-H-A-P-E, just like, you know, our shape, right? Getting in shape. Can everybody can see the messages, right?
Yeah.
Okay, good. Yeah, so go to Athira's website, go to the clinical trials. Then there's going to be a questionnaire for you to fill out. You could do it, or you could have your friend's neurologist do it, but it doesn't have to be a neurologist. You can do it, and they're going to ask you some questions that kind of are a preliminary screen. If you pass the preliminary screen and you're interested, you will have to go to Seattle. And there they will give you a much more detailed evaluation. If there's no cost to you, they'll wind up covering all costs, transportation, everything. So once you go, then you'll be assigned. You won't know whether you get placebo or drug. That'll never be revealed to the end of the trial. But then you would be in the trial. So that's basically the steps. Go to the site, go to the clinical trials, pull down, fill out the questionnaire, probably in consultation with your neurologist. If the clinical team will review that, they'll get back to you and say, hey, you look like a great candidate. Why don't you come over to Seattle? We'll do a more detailed evaluation. Yes, you're in the trial. So that's pretty much how it works. Okay, let me see what other chat things were here. Let's see here. So the question is, when patients return to their near normal after 8 days, does it matter how advanced their disease is when the treatment started? Basically, we picked people with moderate disease for this trial. We didn't pick people for advanced disease. Honestly, I think it will work on advanced disease. And that's because of anecdotal data. There are a lot of physicians out there who are illegally using some of the early forms of this drug. And I am in contact with them, some of them using with advanced patients. So I suspect that it's going to work with advanced patients. So you might say, well, why didn't we do advanced patients? Again, if this was a small trial, we had to make the people as similar to one another as we could. So we had a certain criteria for these people. And it was kind of late, early to moderate disease. And you might say, well, why would you pick that? If you look at how Alzheimer's progresses, and I know you guys can't see me when I'm doing this, but it progresses very slowly at first, right? And then all of a sudden you reach a period and it goes nearly vertical, right? This is the moderate. And then at the top, it flattens out again. So it looks like a sigmoidal curve. If you want to see an effect, you want to be on the rising phase of the sigmoidal curve. You don't want to be in the beginning where it's flat and there's almost no change over time. You don't want to be at the top where there's almost no change over time. You want to be on that portion of the curve. That's where you have the highest sensitivity for your drug. So to answer the question here, these were all moderate people on this trial. Let's see, I must be missing one more here. Is the company working on patients? Absolutely. Or that was at patents. We have multiple patents. We have a whole legal team that does nothing but patents. So yeah, so patents are, it's an unfortunate thing, but we need patents to be able to get funding, right? Most people are not magnanimous and give us money just to do this, although a lot of people do. Almost all of our initial investors were what I would call philanthropic investors. They had a personal reason for wanting this to work. And even some of our big shareholders, they're not truly philanthropic, but they have personal reasons for this to work. I mean, if you think about neurodegenerative disease, who doesn't have somebody in a family or a friend? There's no one. It encompasses everyone. Mm-hmm. So, so anyway, yeah, it'd be nice if we didn't need patents, so it could just be, available to everyone, but it would never get done if we did that. That would be my choice if we could do it that way. Let's see.
There's
Okay,
one last question.
Okay, let me
in the
see.
chat.
Okay, what is that? Let's see. Let's see. Okay, let me get down here.
Do you want me to read it for you?
No, I got it. Oh,
Okay.
Okay. Do the neurons continue to develop if the medication is discontinued, or they develop on their own resulting from some medication? So let me answer that. The data so far suggests that once a patient is taken off the trial, they will start to degrade in about a month. So what we're envisioning for the final treatment regimen is patients will be taking the drug maybe daily for a few months. And then they're probably going to go back to taking it once a week or once every 2 weeks forever. Actually, FDA has come to us and asked us, would we ever consider running a trial prophylactically? They are thinking that everybody over 50 might ultimately want this drug, whether you're diagnosed with Alzheimer's or not. So we'll see. That's something that we're not ready to deal with as a company. The FDA did tell us if this is approved, we have to, this is something you don't even think about as a WSU scientist, manufacturing. We have to be able to produce 2 billion doses a year. Wow. That's what we're geared up to do 2 billion doses a year. So they you have to have all these things in a line before the FDA approves. It's not just, oh, it worked, but can you actually supply it to anybody, right? It doesn't matter if it, it's like going to a store and they have a sale and you go there and the sale item, there's none of them. Well, that sale was no good. So it's the same thing. They want to make sure you can actually provide what you say you have. So let me see, I'm looking here if I got. Are physicians helpful in telling patients about the trials? I think the answer is yes, they are. The vast majority of people have gotten on these trials through their physicians. Yeah, there's a question like a maintenance drug. Yeah, so Sherry, yeah, absolutely. That's what we envision. We envision this as a maintenance drug. It's kind of like, we don't know how much repair can happen, right? And it's probably going to determine on how advanced the disease is. Basically, we're going to blast the brain with the drug, get as much repair as we can get back. Okay, and then, okay, let's keep repairing the brain as fast as it degrades from then on out. One of the things we know about this drug is it does not care how you injured the brain. Every single model we have ever looked at with brain injury, it fixes it. It's, like I said, we haven't really delved into traumatic brain injury, but there's absolutely no doubt in my mind it will work for traumatic brain injury. So that's a story of the drug right now is, I don't know what number we're using, but it used to be MN. You might wonder, what is MN? That was the company's name was M3 Biotechnology. That's been changed to Athera. So now they all have ATH numbers. So the drug that's in clinical trials is ATH 1017.
Joe, this is Nancy.
Yes.
You mentioned you talked about you can't do it without funding.
Yes.
American Alzheimer Association, Parkinson's, Michael J. Fox.
They all provided funding. In fact, if I went down to the end of my talk here, the last slide would be all the funders.
Okay, I was just wondering, because those are so obviously organizations that would be interested, besides, like the American Cancer and some of these others.
I think Athera received the largest single grant from the National Institute of Neurological Diseases and Stroke ever given. Wow.
Congratulations.
And I think it was like $27 million.
Wow.
And we've had funding from Michael J. Fox, then National MS Society. We've had ALS funding. Alzheimer's Foundation has given us a ton of money. Alzheimer's Drug Discovery Foundation has given us a ton of money.
You know
that. Both to my lab and then ultimately to the company. And that money is always useful because it's what's called non-dilutive money. You don't need to sell stock for that, which makes your shareholders really happy because you're not diluting their holdings. So yeah, it's I've learned a lot that has nothing to do with science activity. Not all of it I wanted to know, but I do know now. Like I can't tell you how much time I spent writing patents.
Yeah.
Yeah, I mean, I spent one summer, one of the big patents we had, one of our first patents was over 150 pages. I wrote it. So, and then we just gave it to the lawyers to clean up the claims. It would have cost a fortune to have it done. I mean, it just, that way, it wound up costing $20,000 or $30,000 instead of $300,000.
Yeah.
So when you're a new company, you have to do these things, or you just can't afford to pay $600 an hour to these guys.
Okay. Any other questions, comments?
I am going to just hop to the end here. I want to just thank all the people who did this. I'm just going to quickly pop down through here. Okay, so these, oh, okay, let me go back one.
Okay, go back.
This is the people in the lab who did, this is how many people did this work. I highlighted a few people that were really absolutely essential. But almost every one of these people made an important contribution. They're graduate students, postdocs, collaborators, vet students, you name it. And I'm sure, I know I missed people. Even my daughter is in there, Erin Harding. As a high school student, she has 2 papers. Yeah, she's the one who works, I said worked for Amazon. We were talking before. Yeah, she's a very brilliant girl. So it was nice having her in the lab. So she's a lot smarter than I am. So it was nice. She's like her mother. Anyway, so that's, and then the last was Nancy, here's people that
Yes,
we
yeah.
found.
Oh, Murdoch, yes, yeah,
At Murdoch, they provided us with a $1 million piece of equipment that was essential.
yeah.
Michael J. Fox, Alzheimer's drug discovery. I mean, there's some that aren't on here. This is an older slide. I could have added more, but many people have put their money into this to make it possible. The Office of Commercialization been super great, absolutely super great. CETA, fantastic. So.
Okay, that's great. That's good. That's good, solid money. That's good.
Yeah. Anyway, so yeah, sorry I ran over.
No, that's fine. I think we've all found it very, very interesting and
I'll get rid of my,
remarkable.
I'm going to stop my share here so you can actually see me. Okay.
Okay. Sue or Christina, anyone else have any questions or comments?
No comments except thank you, Joe. How very interesting, and your enthusiasm is contagious. I just wish my brain operated better.
Yeah, it's I mean, it's hard not to be enthusiastic when so many people could benefit from this. I mean, I feel incredibly privileged to actually have had to be able to do this. I really do. I mean, like I said, there was some serendipity involved, and yeah, it's not everybody gets to do something that might be this meaningful. So.
And this
Yeah.
is Barbara Wells, and I would just say my husband was diagnosed with dementia 2 years ago, and this is the most hopeful piece of information I've received. So thank you.
My pleasure.
I do want to say in 2014, Joe did a presentation for innovators. Do you remember that, Joe, over in Seattle? Innovators, and I was part of that. And he filled up one of the conference auditoriums with so many people who had family members, or they themselves were Parkinson's or had Alzheimer's or whatever. And he got a standing ovation when he got done. I don't know if you remember that, Joe, but you did get a standing ovation after
I'll
that.
take your word for it. I don't remember, but
It's okay. But it's just the everyday average person. Like you said, there's not one person who hasn't been affected by some of these diseases. And so it is a fantastic thing that you and Jay have done or are doing. So.
yeah, well, like I said, we feel very privileged to have been able to do this.
Okay. Well, thank you, Joe.
Okay, my
Sue?
pleasure.
Anything else?
Think not. Nice job, Nancy. Thank you, Joe, very much.
Thank you.
Thank you, Joe.
Have a
Thank
great afternoon.